I will try to give as brief an introduction as I can to my colleague, Bob Weinberg, but it's hard. Bob has been at MIT since 1960, and apparently just can't wait to take over. I was going to tell you all about his PhD thesis, which, by the way, was on small nuclear RNAs. Which still are interesting, and go through a number of wonderful and really moving times as our labs were near each other, and some of the most wonderful discoveries happened next door. Bob's on.
Thank you for that kind introduction. As David implied, I'm a bit of a stick in the mud. I was in this room already September of 1960. I went away for a brief period. When I did a postdoc at the Salk in the winter of 71, 72, Salvador Luria came by, and he told me, he didn't ask me, he told me I would be a member of the then MIT Cancer Center, about which at the time I knew nothing. He didn't really ask me, he just told me, I pretended, having a modicum of pride, faint interest, and so I waited for 2, maybe 3 days before I agreed to come. And it's a decision I've never regretted, but of course here I am now, 38 years later. In 72, I spent quite a bit of time walking through the gutted remains of what had been the Brigham chocolate factory, and began to see the outlines of our future home, which I would occupy for a decade before moving over to the Whitehead, where I've been for the last quarter century. And what's been remarkable for me over the ensuing years is the fact that there have been intimate daily contacts across both sides of Main Street, which have held together a biological community which has been remarkably interactive in spite of the potentially disruptive effects of living and working in different buildings. To my mind, this continued interaction has really been one of the glories of the MIT biology department. Nonetheless, you didn't come to hear me reminisce, and we've been enjoined from doing so, so I'll move ahead to discuss the science. By the way, one of the aspects of this symposium, which I find most remarkable, is how different it is from one comparable symposium of 10 and 20 years ago, because here you've heard with ever-increasing frequency how we're in focus now for the first time on really trying to treat people. A promise that was implicit with Richard Nixon and Ted Kennedy's War on Cancer of 1971. But only now, many decades later, are we beginning to deliver on that promise, or at least attempting to do so, and I think that's most exciting. My own research in recent years has been focused on trying to understand some of the later stages of human tumor pathogenesis. Here is the description of the multi-step process that leads to the formation of the primary tumor, as first enunciated by Kinsler and Vogelstein more than 2 decades ago. And this last stage of metastasis has been one which has been and is still very poorly understood, but it deserves great attention because metastases are ultimately responsible for 90% of cancer-associated mortality. And we don't really understand how they occur, although there are beginning to be some insights. Do additional mutations need to accrue to cancer cells in order for this last step of malignant progression to occur? And can we begin to make a laundry list of those determinants which are responsible for dictating the malignant behavior of cancer cells? In fact, the actual dissemination of cancer cells and the formation of metastases is itself a complex multi-step process. Here we see the multiple steps of this, which result ultimately in the formation of macrometastases. I believe it was Louis Chodosh who mentioned previously that this last step is actually the least efficient of these steps, even though there may be hundreds to thousands of micrometastases in the bone marrow of, for example, women diagnosed with breast cancer. Only half of those who have those micrometastases will ever go on to progress to metastatic disease, a testimonial to the fact that the adaptation of cancer cells, of disseminated cancer cells, to a foreign tissue microenvironment is fortunately a very inefficient process. If one examines this invasion metastasis cascade, as described first by Fiedler, one comes to realize that it's biologically as complex as the steps that preceded it and led previously to the formation of a primary tumor. And that begins to beg the question of how cancer cells are clever enough to acquire all of these multiple cell biological attributes, and whether additional mutations are required in order to empower a cancer cell to successfully execute all the steps of the invasion metastasis cascade and to generate a macroscopic metastasis. In my own laboratory, much insight has come from studying tumor xenografts, in this case experimentally transformed human breast cancer cells. Here you see them growing in mice. Here you see the epithelial cancer cells stained for the epithelial marker cytokeratin. Outside you see the recruited mouse stromal cells, and the green cells here along the edge are of human origin. These cells have shut down their epithelial cytokeratin marker, which is in red, and instead express vimentin, which is a mesenchymal marker. And this shift in their expression of these cytoskeletal markers indicates that these cells have undergone an epithelial mesenchymal transition. As I'll indicate shortly, this involves far more than simply the change in cytoskeletal protein expression. Here one can see something about the topological localization of these cells. Here's a tongue of these epithelial cancer cells, surrounded once again by recruited mouse stromal cells, and if one looks carefully, one sees that the carcinoma cells on the outside of this tongue of cancer cells have shut down the epithelial marker cytokeratin, and instead, over here one sees they now express vimentin. And this indicates, or it certainly strongly suggests, that these cancer cells, when they're closely opposed to the nearby mouse stroma, have undergone this EMT, and to the extent that this is an important process in dictating the future biological behavior of these cells, this begins to suggest that the microenvironment of a cancer cell is in fact an important determinant of its behavior. And this microenvironment cannot be really discerned simply by sequencing, to state the obvious, the genome of the cancer cell. One must also know the cells that are dictating its localized environment in the tumor. In fact, the EMT, the epithelial mesenchymal transition, is a program of ancient vintage. It was developed already in the Cambrian era, and a diverse array of metazoan phyla employed in various stages of embryonic morphogenesis. Here one sees in the sea urchin the cells that have delaminated from the outer epithelium, have invaded into the blastocele, and these cells are going to be the cells which are the unlagen of the endoderm or the mesoderm. And there's a series of other such steps in embryonic morphogenesis, including here the emigration of cells from the primitive chordate neural crest. These light blue cells will ultimately insinuate themselves into the skin, where they will form the melanocytes. And I also mentioned in passing, and just by way of introduction, that the EMT is a multifocal, multifaceted program involving changes in motility and invasiveness in cell surface proteins, as well as intermediate filament proteins, which represent, in many respects, a profound, almost total change in cell biological behavior. The EMT can be programmed, can be choreographed by any one of a series of 6 to 8 transcription factors, each of which can act pleiotropically in an appropriate cell type to program the EMT. And if one ectopically expresses such a transcription factor in an epithelial cell, then often one observes the total panoply of EMT-associated phenotypic changes. Moreover, and conversely, if one takes mouse breast cancer cells, as Jing Young did here, which express high levels of one of these factors, the twist transcription factor, one sees that these cells will metastasize and form multiple metastatic nodules here on the surface of the lungs of a tumor-bearing mouse. But if one deprives the cancer cells of expression of twists through the artifice of an siRNA, then one sees a virtual loss of all metastases, and the few metastases that continue to form give evidence of continued twist expression, never having been deprived of it in the first place. Experiments like this persuade one that in this case, this embryonic transcription factor is essential for the ability of these cells to disseminate. However, whether a transcription factor like twist can on its own convert a fully non-metastatic cell into a metastatic cell remains unproven to this day. Nonetheless, knowing the pleiotropic powers of such a transcription factor, one can begin to ask the question of how many steps of the invasion metastasis cascade could in principle be programmed by such a transcription factor. And here I make the speculation, I think it's increasingly plausible, that if a primary cancer cell turns on the twist transcription factor, all of these steps of the invasion metastasis cascade are within its purview, save for the last step, which involves ostensibly the adaptation of cells from one tissue to a tissue microenvironment, a strange tissue environment, which to my mind is not within the purview of such a transcription factor. If this kind of scheme is further substantiated, it represents a great conceptual simplification of metastasis, because it indicates that by turning on one of these transcription factors, the whole processes involving the metastatic, the physical dissemination of cancer cells can be controlled and can be activated. Nonetheless, the last step, the one of colonization, still represents terra incognita. We really don't understand how this occurs. Importantly, if this is all true, then in fact the activation of this such a transcription factor can be achieved without any genetic alterations in the cancer cell. And the work of Vogelstein and Markowitz has demonstrated recently that in the case of colon cancer, they estimated it would take 18 years or so for primary colon carcinoma to form. However, the metastases in the liver were already apparent within the next subsequent year or two, and that's consistent with the notion that this last step of dissemination really does not seem to depend on additional rare mutational events. It doesn't prove it, however. In my own laboratory, we've investigated a series of alternative transcription factors. Here's the work of Piyush Gupta, who transformed human melanocytes using a transformation protocol that Bill Hahn had developed. He had known that a whole variety of normal human cells can be transformed by this protocol, yielding tumorigenic, vigorously growing cells, which rarely or ever metastasize. In contrast, putting the identical set of transforming oncogenes into normal human melanocytes, one got metastases by the hundreds in the lungs, the liver, the spleen, and the mesentery of the intestine. And this tells us a couple interesting lessons, one of which is that the cell of origin in which transformation takes place, prior to any kind of acquisition of somatic mutations or experimentally introduced mutations, the cell of an origin is an important determinant of the eventual metastatic behavior of the cancer cell. In the case of the melanocytes, as I mentioned before, they derive from the primitive neural crest. They employ the slug transcription factor, which can induce an EMT. And indeed, if one looks at experimentally transformed melanocytes, they express 1,000-fold higher levels of this transcription factor than do breast cancer cells. Once again, indicative that the normal differentiation program is an important determinant of behavior subsequent to transformation. Gupta's work showed that by shutting down slug, in this case 93%, he got a 93% reduction in metastases if the transformed melanocytes were deprived of this transcription factor. And this represents a nice vignette where the early embryonic program that allowed the ancestors of these melanocytes to disseminate throughout the normal embryo is employed and resurrected by the melanocytes if they undergo transformation in the adult. In fact, there are a whole series of these transcription factors, and the work of Sendhil Raimani has begun to generate interactomes in which these different transcription factors interact in different ways. It's complex, but it seems increasingly likely that they work in different groups in different kinds of human carcinomas. It's plausible now, but hardly proven, that there are several of these transcription factors that are active in virtually all human carcinomas, but we're far from being able to prove that experimentally. In the case of Mani's work, he was interested in the consequence of creating an EMT. As the title implies, the obvious target of the EMT is a mesenchymal cell, but for reasons that I don't have time to enter into now, he speculated that the product of the EMT was actually a stem cell, a speculation that earned him the derision of his lab mates, since it seemed just too sexy and convenient that stem cells, which everybody else was working on, also impinged on his own work. Still, he forged ahead. I never hearing about this till months later. And here I just refer us to the general conceptual scheme that is widely embraced vis-a-vis stem cells. In normal epithelial tissues, one imagines as a self-renewing stem cell, transit amplifying cells, post-mitotic differentiated cells, and that the bulk of the mitotic activity in the tissue involves these transit amplifying or progenitor cells. And this scheme has been appropriated by those who are interested in the organization of solid tumors, such as breast cancers, as Michael Clark and Mohammed Al-Hajj first showed. In this case, the self-renewing stem cell is a cancer stem cell which has the ability to seed a new tumor when it's transplanted into an appropriate host. The great majority of the cells in the tumor, although they may be genetically identical with the cancer stem cell, have, because of changes in their transcriptional program, given up the ability or the opportunity of seeding a new tumor, even though they may constitute the bulk of the tumor mass. And this scheme also holds implications, to the extent it can be verified, in the whole problem of metastasis. because this cell, if it successfully disseminates from a primary tumor, is qualified to create a metastasis because of its self-renewal potential. The great majority of cells in the primary tumor, however, according to this scheme, even though they may successfully disseminate, are not qualified to seed metastases because they have given up the option of self-renewal. In the case of Amani and Wenjun Guo, who worked on this, they used the CD24 and 44 markers that had first been illuminated by the work of Michael Clark. And here, just by way of orientation, I'll show you that the FACS analyses they used demonstrated that according to the previous work, the position of CD24 high, 44 low, represents the position of non-stem cells in this FACS analysis. Up here is a position of putative stem cells, 44 high, 24 low, converse cell surface markers. Of some interest here is the fact that these cells were propagated in cell culture. And to the extent that these are bona fide stem cells, this begins to suggest that one can actually maintain stem cells in tissue culture rather than just in the specialized microenvironment of a stem cell niche, as is widely believed. In the case of Mani and Guo's work, when they forced cells through an EMT through long-term culture with TGF-beta, which is a rather ineffective way of doing so, the cells underwent a morphological transformation from an epithelial to mesenchymal phenotype, but more importantly, they migrated en masse from the non-stem cell position up to the stem cell position. And this could be recapitulated by ectopically expressing in these immortalized human mammary epithelial cells either the snail or the twist EMT-inducing transcription factor. In both cases, one observed an en masse, we have to stop. In both cases, he observed an en masse migration from a non-stem cell to a stem cell position. And this began to suggest, once again with morphological transformation of cells, that the cells move from the non-stem cell to the stem cell position under the actions of these two EMT-inducing transcription factors. In the end, however, a more persuasive demonstration came from looking at the naturally resident majority population of non-stem cells and stem cells, comparing the relative levels of expressions of various messenger RNAs in these two kinds of tumors. And in the putative stem cells, one found 1/200th as much of the epithelial marker E-cadherin. Conversely, the putative stem cells expressed between 40 and 100-fold more of fibronectin, vimentin, and N-cadherin, all mesenchymal markers, and comparable overexpression between 30 and 120-fold of 4 EMT-inducing transcription factors, FOXC2, CYP1, twist, and snail. Slug, in this case, was not significantly overexpressed. Still, here is a naturally existing affiliation between the expression, the endogenous expression of an EMT-inducing transcription factor and residence in the stem cell state. And so one gained confidence that there really is an intrinsic natural affiliation between stem celledness and the actions of these EMT-inducing transcription factors. This could also be extended to, for example, looking at normal human mammary epithelial cells from reduction mammoplasties. In this case, a relative 150-fold overexpression of the CYP1 EMT-inducing transcription factor. And reassuring was the observation that the putative stem cells looked mesenchymal in monolayer culture, and they could form mammospheres in three-dimensional culture. These mammospheres are known to contain mammary epithelial stem cells from the work of a number of laboratories. Conversely, the non-stem cells have a cobblestone phenotype in monolayer. They fail to form mammospheres with any efficiency, as you can see from these two bar graphs. Induction of either snail or twist has a profound effect, as well as long-term exposure of TGF-beta, in increasing the number of mammospheres in 3D culture. I portray this mammosphere assay as a surrogate assay for stem celledness in vivo, as it is widely used in the mammary field. In the work of our collaborator, Nelly Polyak of the Dana-Farber, she looked at both human breast carcinomas as well as normal human mammary epithelial cells and discovered, as you can see here from this heat map, a preferential association of mesenchymal markers with the CD44 high, 24 low stem cell fraction. And I mention this for no other reason to demonstrate the fact that the more we look, the more we see that there are very strong parallels between the stem cell program of normal mammary epithelial cells and the stem cell program of breast cancer cells. It is not as if the breast cancers invent an entirely new stem cell program. Instead, they seem to appropriate it almost entirely from the pre-existing normal mammary epithelium and to exploit it for their own internal organization. Taking all this together, this persuades one that the EMT program is actually dangerous for two reasons in terms of clinical tumor progression. First, it empowers cancer cells to physically disseminate, and secondly, it enables these disseminating cells to be self-renewing, which I argued before, without showing it to you, that this was a prerequisite to successful metastasis formation, and that in the absence of self-renewal potential, a disseminated cancer cell would have a very hard time, if not an impossibility, of forming a macroscopic metastasis. In the end, however, we wished to create more persuasive biological proofs that there is really an intimate connection between the epithelial-mesenchymal transition and the stem cell state. And here I refer to the work of Wenjun Guo. We're switching now from the human to the mouse mammary system, and here the FACS analyses look different. Here are the stem cells, as he showed, and the non-stem cells, the work of John Stingle and Jane Visvader being very influential here. And here one also makes use of cleared mammary stromal fat pads. The fact is, as he showed here, if one takes these putative stem cells and implants them in a cleared mammary stromal fat pad of a mouse, after 6 to 8 weeks, these putative stem cells, which look mesenchymal in monolayer culture, generate entire mammary ductal trees which are intact, save for a connection to the nipple. Conversely, the putative non-stem cells fail to form such a mammary epithelial ductal tree upon implantation into the mammary stromal fat pad. And of course, as you would imagine, because they're non-stem cells, they form an epithelial cobblestone-like monolayer. Moreover, Wenjun Guo found when studying using RT-PCR the expression of a series of different transcription factors that, for example, the slug transcription factor, which I alluded to before, is expressed 36-fold more highly in the putative stem cells rather than the non-stem cells. And upon doing immunofluorescence, he found that slug is indeed expressed along these abluminal cells, which are the putative locations of mammary epithelial stem cells, in this case, in the mouse mammary gland, this being a cross-section of a mouse mammary duct. And so he undertook to examine the question of whether he could impart to mammary epithelial cells an increased stem celledness using this mammary gland implantation assay. Indeed, if he could impart to cells an ability to reintegrate into the mammary stromal fat pad and to regenerate an entire mammary gland, this should, by all rights, be a very convincing demonstration that the implanted cells had acquired a stem cell function. And so he took two populations of mammary epithelial cells, in one in which he induced the expression of the slug transcription factor reversibly for 1 week prior to implantation into the cleared mammary stromal fat pad. Another population of control cells, one was labeled with RFP, the other with GFP, was not so induced. He mixed these two in equal numbers together, and then he asked them to undertake a horse race to see which ones of these populations of cells would be more successful in engrafting and generating the mammary ductal tree. Keeping in mind that the exposure to slug occurred prior to introduction into the mammary stromal fat pad. That is, it occurred ex vivo. After 1 day, the two kinds of cells, red and green, looked yellow, and they were indicating they were present in roughly equal amounts 1 day after implantation. After a week, there began to be a slight prevalence of the cells that had been exposed ex vivo to slug, and after 6 or 7 weeks, now the cells that had been exposed ex vivo formed these ductal trees, whereas the cells that had not been showed minimal ductal tree generation. This orange here being testimonial to the fact that even normal mammary epithelial cells have a small number of stem cells within them. Nonetheless, data like this persuade us that there indeed is much to be said. Indeed, we are convinced that forcing cells through an EMT in vitro shows a greatly increased rate of stem cell formation. We could demonstrate this as well with the mammosphere assay through the ectopic but reversible expression of slug prior to inception of the assay. And equally interestingly, if you took mammary epithelial cells, normal populations of mammary epithelial cells, and deprived them of slug expression prior to putting them into three-dimensional culture, then this resulted in their loss, essentially their entire loss of stem cell activity. And so we speak with increasing conviction that there is indeed an intimate connection between passage through an EMT and the acquisition of stem celledness. Whether this is the only way by which cells, epithelial cells in the mammary gland and potentially elsewhere, can acquire stem cell function is something that we still don't know. Now, going to the issue of therapeutics, which I said is now increasingly the focus of our cancer symposia over the last decade, we were aware, and I believe Louis Chodosh mentioned this already, that in fact many existing therapies result in the elimination of the non-stem cells in a population of treated carcinoma cells, allowing the survival of the stem cells, which are more resistant to therapy. And here one sees this in a schematic way. Here's a population of cells within a tumor. Here are the stem cells and the non-stem cells. As one has observed in several different human carcinoma types, treatment with a variety of conventional therapeutics results in the ablation, effectively, of the non-stem cells, in the collapse of the overall tumor size, i.e. debulking, but the survival of, in fact, a population of stem cells which live to the day when they can begin to regenerate the tumor once the therapy is lifted. And so in order to address this, Tamer Onder produced a population of cells which he had placed constitutively in a mesenchymal state through the shutdown of E-cadherin. And here I just give you indications of this through the acquisition of Vimentin, for example, and N-cadherin. And then he and Piyush Gupta began to examine whether these cells, which had been made mesenchymal and thus having a stem cell phenotype, might also be used as reagents with which one could detect compounds that could preferentially kill putative stem cells rather than non-stem cells. And the great bulk of this work then proceeded not in my own laboratory, but in the laboratory adjacent at the Broad Institute of Eric Lander and in the hands of Piyush Gupta. And here I describe his work, in which they first began to examine the effects of two conventionally used therapeutics, doxorubicin and paclitaxel. In this case, I bring your attention to the fact that the blue represents the behavior of non-stem cells, non-cancer stem cells. The red represents the behavior of cancer stem cells that have been forced through an EMT. And here, looking at increasing drug concentrations, one sees, as advertised, that the non-stem cells with these two conventional therapeutics, the non-stem cells die or are killed off at about 1 order of magnitude lower drug concentration than the putative stem cells for both of these reagents. Piyush Gupta then proceeded to screen 16,000 compounds in the library that they had then at the Broad for those compounds that might preferentially kill the putative stem cells. And here they found 22, of which 2 were examined in detail because of the availability of the drugs. Salinomycin and abamectin, they're both used as antiparasitics, actually in animal feed. So the next time you eat a hamburger at McDonald's, you may be ingesting a wee bit of this. It's rather innocuous, salinomycin, but just to return to our theme, it is able to kill off the cancer stem cells at about 8 to 10-fold lower drug concentration than the non-stem cells. So it acts in the converse way. And here, I give you more evidence of that. Here is a population of these breast cancer cells, 95% in the non-stem cell position, 5% in the stem cell position. If one treats them with paclitaxel, a Taxol analog, and one greatly reduces their overall number. But among the surviving cells, 70% now are stem cells, and the remaining 30% are non-stem cells, in consonance with what I mentioned before. Conversely, salinomycin resulted in the presence of only 0.2% of the surviving cells having the stem cell antigenic phenotype, with the great majority, 99.8%, having the non-stem cell phenotype. So none of us is interested in portraying this as the answer to cancer in terms of this being a precursor to drugs that might be clinically useful, if only because the formation of clinically useful drugs is extraordinarily complex and dictated by a whole variety of parameters that we're not equipped to deal with here. What I mention this only to indicate the fact that one can, with relative facility, screen for compounds that can preferentially kill cancer stem cells rather than non-stem cells. Still, one can pose the question, is this really the answer to cancer? In other words, by getting rid of the cancer stem cells, will one indeed succeed in getting rid of tumors? And here I refer to the work of Christine Schaffer. She was working with immortalized human mammary epithelial cells, which are essentially identical biologically to normal human mammary epithelial cells. And she discovered there were floating cells in the Petri dishes. Now, we all know in this room here that floating cells are dead and should be dumped. But she didn't know that, so she took these cells And she put them in another petri dish, and lo and behold, they re-adhered and started growing vigorously. She called these cells flopsies, for floating populations of cells, and discovered that they had stable phenotypic differences from the parental population. Morphologically, they're different. They express different cytokeratins. They express greatly different levels of cell surface, epithelial surface antigen, and they have a much higher proportion, naturally, a naturally resident subpopulation of the CD44 high stem cell-like cells. And they maintain this phenotype in culture rather stably. Of some interest is the fact that she generated then single-cell clones from these flopsies, these floating populations of cells. For example, single-cell clones that are CD44 high or CD44 low. Here are some of these clones. Interestingly, all of the populations had a minority population with a stem cell-like immunophenotype, and that suggests the possibility that if one looked hard enough in the petri dish of all epithelial cells in culture, it's possible that there are stem cell-like cells that naturally reside as subpopulations of propagated stem cells in vitro. Again, I make that as a speculation rather than as a proof. Clearly, these cells behaved in one respect like stem cells. Here, the CD44 high cells generate CD44 low, more differentiated cells. And this is in consonance with our preconception that stem cells should differentiate to yield non-stem cells. She followed the fates of these various single-cell clones over a period of 12 days. And what she discovered was, as I just mentioned, the CD44 high cells generated CD44 low cells, these little red dots here. And that was indeed anticipated. But conversely, she did not anticipate the following, that the CD44 low cells, the non-stem cells, with the passage of time generated an increasingly large subpopulation of CD44 high stem-like cells. And clearly the most direct explanation of this is that there was a contaminating subpopulation of stem-like CD44 high cells from the very beginning of this experiment. But this explanation is embarrassed by the fact that, in fact, the stem cells actually proliferate more slowly than the non-stem cells. And it makes it hard to argue that the reason why this population emerged was that these red cells progressively outgrew the larger population of non-stem cells. And this has been done time and again. Another way of addressing this is to mix CD44 high cells with CD44 low cells in equal numbers. The orange cells are CD44 high initially, the black cells are CD44 low. Follow them over a period of 15 days. The proportion of the initial CD44 high cells in the population of tissue culture cells as a whole goes down progressively because I just advertised that they grow more slowly than the non-stem cells. Conversely, the CD44 low cells begin to generate an increasingly large proportion of CD44 high cells. So we believe that this is actually a process that occurs naturally in the petri dish, and its frequency depends on the state of the cell. The parental population generates these CD44 high cells very slowly, but with increasing introduction of transforming oncogenes, the rate with which the CD44 high cells are generated spontaneously in culture increases. Here's her collaboration with Paul Wiggins, in which they calculated that per cell generation, cells which just have telomerase in them generate, per cell generation, this many CD44 high cells. If one puts in the SV40 early region, that one doubles the rate of this spontaneous generation, and if one puts in the RAS oncogene on top of that, one doubles it again. So one can perturb the rate with which this occurs spontaneously in culture, and moreover, if one looks at these various populations and transforms them subsequently through the introduction of other oncogenes, one discovers, for example, that the CD44 high cells that are initially CD44 high prior to transformation now generate tumors which have as many as 300 or 400 metastases, whereas the parental cell population that did not derive from flopsies but has the same set of introduced oncogenes generates no such metastases at all. This, by the way, is also a rather stark demonstration that the differentiation state of normal cells prior to their undergoing a neoplastic transformation is a very important determinant of the metastatic potency post-transformation. Moreover, if one transforms the CD44 high cells and then looks for the frequency of tumor-initiating cancer stem cells, it's actually about 1,000-fold more concentrated, the tumor-initiating cells, than if one transforms the parental population. So once again, there's a strong influence of the pre-existing differentiation program on the subsequent behavior of the resulting neoplastic transformed cells. The CD44 high cells, as one might anticipate, when implanted into mouse stromal fat pads, these are human cells, so they form only these minimal ducts, but nonetheless, these are indicative of stem celledness. If one takes all this together, that is to say, these observations of Christine Chaffers of the spontaneous formation of CD44 high cells, if one then assimilates also the notion that the stromal cells seem to induce cancer cells to undergo an EMT, as is indicated here, and the fact that the EMT produces cells with a stem cell-like phenotype and indeed biological behavior, then one comes to realize that in fact this diagram needs to be redrawn, and that there is, depending on the biological context, a spontaneous dedifferentiation, and that there's much more plasticity than one might have imagined in these populations. And this, and it is my last slide, sir. Thank you for that subtle hint. Now, this is ultimately the desired outcome if one were to eliminate cancer stem cells from a tumor. Removal of the stem cells, and then the whole tumor will melt away. But now I would suggest that we are confronting a more complex scenario, because it may well be the case that if one succeeds in eliminating the cancer stem cells with an agent like, for example, seleninomycin, that the surviving non-stem cells will spontaneously regenerate, de novo, new stem cells that will go on to regenerate, in turn, a large and vigorously growing tumor. So this complicates how one thinks about getting rid of stem cells and non-stem cells in tumor therapeutically. The work was done by a group of unusually good-looking people, as you can see here, many of whose names I've mentioned. Again, thank you for having me.